So welcome to our laser scanning microscope from Zeiss. It's called LSM710 and it's a high quality confocal microscope. It's not fast as the other system we have, but it's a solid workhorse system. So you turn it on by clicking these 3 buttons and you see all the hardware goes on. You can see some of the hardware has some tape on it. You don't need to, you should not touch that because it is turned on already. After these 3 buttons, we go to the next door, turning on the laser. So now we are in the next door room where the laser is. So this big box is an argon laser, and you turn it on this on position here, and you can see the diodes go on. And this is the power control and the fan. The actual laser is turned on by the key, and you can also hear the sound when you turn it on. So after turning on the laser, then we switch it to the on position, and then we turn on the real-time PC. So that's a second PC controlling all the parts, and it needs to be turned on separately in order for the system to turn on properly every time. And then last we turn on the PC. After turning on the laser, you need to put this toggle into on position. So now the laser is actually active, and it's run now in normal operation mode. If someone has been touching this button, it goes into red when it's warmed up. You don't want it in the high reduced lifetime position, so always keep it with the green diode on. Moving down to the touchpad control, you can go to load position to put in your sample and be in a safe distance to the objective lens. And you can turn on the transmitted light and the reflected light, and then you can move on to control other parts of the microscope. And this system has 20x objective and 40x, so the 20x is a dry lens. The 40x is a water lens. So for that you need water immersion, and you see that here. So you take a droplet of water and put it on the lens. When you're going to use the 63x oil lens, then you need oil, and we have 2 different flasks for that. So for room temperature, we have this oil saying 23 degrees, and then we have another one saying 30 degrees or above. And you can also control the reflected light path. So the first cube here shows a triple band filter showing DAPI fits trits. The second position is a polarizer, which you need for DIC imaging. Then we have different colors, so there's some single filters for green, red, and far red. This can also be controlled in the software. You can go through and use some other functionalities here, and also turn on incubation, because we have an incubation chamber controlling the temperature, and you can set the temperature here on and off. The rest you will control in the software. Okay, so after all the hardware is turned on, you start the LSM user in the Windows startup page. And you map your network drives, and you start up Zen Black Edition. You will be asked to start the system or use it as an imaging platform. I recommend that you book computer for imaging and not block this system for processing your images. So start the system. And you can click this arrow to see the process booting up. And the only issue you would encounter here is that the real-time controller will not be active. And that could be if you didn't turn on the red switch on the panel. So while turning on the software, we'll start installing the sample. And you can see that we have an incubation chamber here controlling the temperature if you need that. Just start opening it all and taking the top to the side. When they are out to the side, you can take the illumination pillar and push it backwards so you have some workspace. And I'll put my samples in. Please only put the sample in and do not take the stage insert out if you don't need it. Start out load position and put in the sample directly. And when you got it in, you simply move it to the position you want. You may have a reason to change the stage insert. I'll show you how. Just take it out like this. And you can see the red dot fits to the lower left corner where there's another red dot. You put it in and you hold your hands to the side in order not to move the stage manually. Because if you push the stage, it will be damaged, and it's somewhat loose already. So this insert was for slides, and the other insert we have is for plates. So it could be a 96 well plate. Again, the red dot is here, and you put it in and you slide it, holding against with the thumb. Alright, so now the sample is in position and switch back. Right now I don't use the heating, so I just leave the incubator open. I put it back to work position. And for locating my slide, my cells, I go to the low magnification, so I go to 20x. And I move by the mechanical joystick here, move to my sample, and I put the reflected illumination to on position, and I go to the reflector, activating one of the filter sets. Now you can see white light is coming up to the sample, and I'm now detecting 3 colors. And I can look into the oculars and find focus. So now my cells are in focus, and I move to the software. If I want to go directly at this point to 60x, I go to, or 40x, I can go to 40 and remember to add water. I can also go to 60x and remember to add oil. And I'll just also control the shutter here, so the reflected shutter is here, so I don't blind myself. And I push it back, take the sample away, and then I add oil. And oil is added like this. Take this up and leave the first drop inside the flask, and just put a small drop on top of the objective lens. And refit the sample. And press done. And again, reflected light is activated. And I find my focus again. Here we go. And remember to shut the reflected light whenever you are in focus. So I'll show you how to install your sample, and please, if you are just using slides, keep this slide insert in the system always, and just from the load position you can directly put in your slide with a cover slip downwards, because it's an inverted system. So now at this point I am at 20x magnification, and I want to now turn on the reflected light, and you can see that probably bright light in the system now, and I find my focus. Once doing that, I may want to go to a different objective lens, so I now press 40x, and this is a water immersion lens. If I want to use that, I would add water. Now I push the 63x, and I'm just shutting the reflected light, removing the sample, and then I take my immersion media, and the first drop I leave inside the flask, and then I just put a small drop on top of the lens. You see now a bit of oil, and I put it back like this, and I go to work position. And again I turn on my reflected light, and I find my focus. Now that's done, and I'm ready to go to Confocal, and I shut off the reflected light in order not to bleach my sample, and then I go to acquisition mode in the software. Okay, so if you need to go from analyzing slides to plates or whatever else, then you may need to take this out. And the way to do it is to remove it, just clicking up, and when you want to set it back, then put it in this position before these springs and firmly put it back again. So slide it in this way. And while sliding, you should hold your hand so you're not moving the XY stage manually. You can also use a different insert which is on the shelf. And you can see here a red dot should be in this corner with the other red dot, and you slide it in the same way. This insert fits to a plate. You can see you could put a 96 well plate or whatever you want to image and fit it directly in there. So when you want to install DIC in this microscope, it's manual. So the first thing you need is the prism needs to be inserted below the objective lens, and this is a prism for 63x, and it has DIC number 3. The same is up here, and the condenser you can see DIC 3, so this is the position you want to be in, and the prism is installed as default in this system, but now you can see how to install it. So put it below the lens and switch it in. Then when you need to install or to adjust the correct DIC illumination, you would first need correct curler illumination, so you go to 10x illumination and adjust the height of the condenser. You insert the DIC prism you did and the slider, and then the polarizer. So this is a polarizer, so it's slided in also as default in the system, and then you simply turn on the transmitted light, and you go to the objective lens you want to work with. So turn on the light in the software, and also open the shutter either in the software or here, and then you start after you focus, you can increase the you can change the polarizer and also the position in here of the DIC, and these two in combination you will find the right DIC position. So after you insert all the correct inserts, you will also need to decrease the aperture stop, which is this small wheel down here. When you do that, you increase the contrast in the DSC image. When you start the computer, you see this script and you use it for mapping your network drives. You type in your AU ID and your AU password, and then you are able to see your drives, so your O and the U drive. You can save all data to the local D drive, and then you can move it when you're done imaging to your network drives. And here you see the software Zen. You simply open it, and you see here the process. And in case you forgot to turn on a switch, it would usually be the real-time controller. It will simply go no further than this step. So make sure you turn on everything, and then you should see these steps, and the software starts up properly. And in the upper right corner you see a zoom function, so you can adapt to what fits your eyes. And in the left part you see locate, acquisition, processing, and maintain. We'll focus on locate and acquisition in this video. And locate, you start out turning on the light you need for the microscope. It could be transmitted light when you want to look at tissue as a sample with a normal white light, or reflected light, which is fluorescent light source. So you turn on the light sources up here. In this point you can make a configuration, so you can make it fit to what you need. But here you see the microscope control, so basically a scheme of the microscope. In the upper light source it's a transmitted light. You tune it, so you give it a percentage, and you shut the shutter and open it. And down here you have the stage control, so you're moving around with your sample. And the focus, so the objective lens is going up and down. And this is your specimen. And the lens. So you can see here we have 20x dry, 40x water, and 63x oil. I'll start out with 20x just to find focus and start orienting myself on the sample. And the light source is turned on and the shutter is open now, but there's no cube in, so I will add a filter cube here, and I can use the blue, which is actually a multiband filter. You see the blue, green, red. You can also choose the analyzer, which is for DIC imaging. It's for polarized light. You can also have green, red, or far red. Far red you cannot see by oculars, so it doesn't really make sense here. And in the bottom you see the incubator, so you can turn on or off the heating and select the temperature you need. I will not use the incubator right now, so I turn it off again. And my sample is in focus, so I will start out in acquisition tab, and from top to bottom you see here quite importantly that you can tick on this show all tools, and when you do that you see more information, so you can handle everything in the microscope. And the smart setup is the first button you see, so you can select your dyes. So it's a help feature that makes it easier for you to set up your experiment. Here I select Höchst and Sci5, which is the two colors I have in my sample. And you see here you can add recent, or you can search for something. Let's search for Texas red, and then it comes up here. In my sample I only have 2 colors, so I can show you the difference here in the proposals. The first is the fastest. You will combine the 2 channels in one channel. I add also here the EGFP, so you have blue, green and red. And in the first you see they are all combined in one track. In the second they are split into 3 tracks, which separates them perfectly and you get the most signal. However, it takes 3 times longer. You can see the tails of all signals is included here, so you get a higher percentage. In the smartest to the right is usually the smartest, so you trade off the speed and the signal, and in this case you have 80-90% signal, and your speed is still fast. For this actual sample I have only 2 colors, so I'll just show you with the smartest feature to the right, combining into the same track. And press apply. And let's get started. So you'll start seeing some images here with the 20x dry lens. So it starts scanning automatically. And here I'll show you the different parts of the microscope and the features you have to adjust before you get going. Up here you can do autofocus and you can do snapshots, and in these buttons down here you get more advanced features I can show you in a minute. The first tab here is the laser. You can see which kind of laser we have. The first is the argon laser, controlling 3 laser lines. That's a big box in the next door room. If you don't need these laser lines, please don't turn on the argon laser. Then you have 405, 543 and 633. So the 405 is a diode laser, and the 2 long wavelengths are gas lasers, helium neon laser. And the next thing is actually what you already did in the smart setup, and this is showing the protocol. So we are now acquiring in one track we are acquiring 2 channels, blue and a red, and you see them here, and you can fine-tune. If you're not happy with this, you can select only what you need from the 2 channels. And that will give, of course, make sense. If you had a green channel in between, then you may need to cut off the tails of the signal in the blue and the red. You see here channel 1 is the blue, channel 3 is the Psi5, the far red. And in the bottom here you see TPMT functions. That is the transmitted detector, and you can activate it, and then you add another channel together with the fluorescent light. And if you do not in the top show all tools, then you don't see this function at all. So it's important to show all tools. Here you see it again. Next, in the acquisition mode, you set the objective lens as a first, and we started out with 20x just to find our sample. And then I'm moving to higher magnification. It could be 40 or 63, and 63 is an oil lens, so I go back to locate and add oil and find my focus. And I again need to turn on the shutter and the filter, and I am now in focus, so I want to image with this high magnification lens, and I want to scan within a frame mode, so I want to have a XY image, not a line scanner. And yeah, you can have line, spot, or frame. And the frame size I want to start out with is a low or medium resolution. I usually do 512 by 512 in order to set the settings quite fast, and then I increase the resolution when I do actual images. So here you see some presets, and pressing this optimal, then you see the optimal frame size when using this lens is 1912 by 1912. But I said I go lower to do my settings. And the first thing is then to adjust the speed. And usually the system starts with high speed, and you can hear the scanner using this speed 9. You get a pixel dwell time of a bit more than 1 microsecond. If you reduce the speed, yeah, you can see if you increase the speed to the level 10 or the maximum, then you are almost down to 1 microsecond, which also gives some noise in the image. Of course, you may have reason to go fast, but you'll get better images going a bit slower. So keep it at 7 or 8 will give you a better image quality. And this pixel dwell time of 3 microseconds. Next function is averaging, so you can acquire, in this case, 1 or more images, and if you acquire more images and average them, you will cancel out some noise in the images. So let's say you acquire 4 images and you have noise in only 1, then that noise would be averaged out. So you improve the image quality. Of course, you also acquire, or you spend, in this case, 16 times the time acquiring, or you should maybe start with 4 time averaging, and that will give you a quite dramatic effect from doing no averaging to 4, and going up to 16. It's not so much of information. So bit depth, you can choose 8 bits, which is what we can see by eye, and that's 2 to the power of 8. Number of grayscales in the image. So if you only want to use it for imaging and image presentation, you have no reason to go higher. But if you want to quantify, you should go to a higher bit depth, and you may have a protocol going for 12 or 16 bits. And usually going to 16 bits, you'll get more details in the image. So you have 2 to the power of 16 grayscales in the image. Next thing is the scan area. So maybe you only want to acquire a few cells instead of 10 or whatever could fit into the field of view. And then you don't want to spend time acquiring too large an area. So you can zoom in using this function, and you can also tilt it, rotate it, like this. Yeah. And now I go back to zoom 1. You can grab the corners, and then you can tilt the field of view if you want to. And here you see our channels. So we have only one track in this case, because we combine the blue and the far red into this one track. And it's activated here. And if you're doing more tracks, remember to switch from track 1 to 2 and so on when you align the system. Now I press live, and live is a fast mode of imaging. So I use it just to set focus and navigate to find my cells. And then I go to continuous. You can see I change from live to continuous. And continuous means that you use the actual settings, the speed, and all what we did above, and you see how the final image would look like. Here I change the laser line 405 laser for the blue, or DAPI. So normally 2 to 3% is the range we use for the laser, and then tune the gain afterwards. That's the sensitivity of the detector. Always take a look at the display or the histogram. You can see here we have a signal in around half of the histogram. If we increase the laser, you can see the signal goes all the way to the right side, so the high intensity of the signal, and we see on the image we see red indications indicating saturation. And we don't want that, so we should go down with the laser and go down to 3%. And we have a good representation here. So going back to Dimension tab, you can toggle between range indicator and the actual color. And in the Display tab, you can set the display, so you can manually, or you can start automatically, then it selects it for you. And you can also deselect automatic and do it manually. So then you can grab this right side of the intensity bar, and then you see much more intensity, even though the actual data is the same. So this is only the display of the data. And it has these best fit and minimum and maximum, so you can present it as you like. Now I stop the continuous mode and move to Sci5 and start again. And back to Dimension to go to Display Only channel number 3. And I see now my red signal. And you see the intensity is quite high, so the gain, which is the sensitivity of the detector, was quite high. Normally that is between 500 to 750, maximum 800. And also you see here the pinhole, so that is what makes a confocal system confocal. So this one area unit is defined as the standard for the confocal, but you can open or close that pinhole to get more light, and also you get more out of focus light if you increase it too much. Now I set it the pinhole as for one area unit, and the gain for 650 approximately. And here I show both channels, so you see both the blue and the red at the same time. And up here you see it in 2D. You can also see it in split mode, 2 different channels and 1 combined. And this is intensity, histogram, a colocalization tab, and so on. This simple image is best represented in 2D. And you can zoom in here. And in the display you can see it for both channels combined. And because it was a bit dim before, you can increase the display here. You can see that done by 2 individual modes. So at this point we acquired a simple 2D image and we want to do a bit more advanced. So ticking on the C-stack, you will have another function shown down here. You see the focus indication to the left, and instead of using continuous here, I use live because that's faster. And I go to my image. And then I want to detect where to start and where to end. So I go down to find my focus. This is about where the sample starts. And I put it as my first position. Then I move up with the focus. And this is where the sample ends. And we can see here in the left, when going up and down, we see a representation of the top and the bottom and the intermediate stacks. And in this case it has an interval of 1, and we would then acquire 5 stacks within this range of 4 microns. And we need to press this optimal to get the best sampling according to Nyquist, and that will end up with 13 slices in this section. You can of course do it manually, maybe select 10 slices and do it the way you want, but 13 is the proper way to do it. And once we have activated one of these functions, it's no longer a snapshot, then it's an experiment. So we start experiment. And you see here, after it's done, you can see in this ruler, you see the C position. So we go through the depth of the image. And again, we can go to dimension to change the display. And in graphics, we can add stuff to the image. And here we see split mode again, and gallery and ortho. So ortho, it is a valuable function for 3D representation. So you can go in X and Y, and then you see the depth in the top and to the right. And going through the stacks here. And you can also select the channels in the lower part. Next function is time series. So starts out with 10 cycles, and you see the tab in the bottom. Down here you can select the number of cycles you want, and also the interval between each cycle. So let's say we wait 10 seconds between each new image would be acquired, and acquire 70 images, for instance, or change it the way you want. You can also do bleaching experiments, which is also a time series. And then you can do tile scans. It starts out with 5 by 5. We can also do something smaller. So let's say 2 by 2. And then we can start an experiment from here. So now it requires 2 by 2, 4 images, and tiles it together. And you see here a larger field of view with 4 images. In the tile view you can do much more complex stuff in the LSM710 system. But here you can also do some positions. So add the positions you're interested in. So I put this 1 position, 2 position, and I move to another area. And then I acquire tiles, because that is activated, and also positions. And you then see in the, before we had the ruler of with the C position, now we have position. So position 1, 2, 3. And you can of course combine C stacks and tile scans, and then you have the option of going to different positions and up and down. Now I selected only the regions tab, and I can add some regions within my field of view. And with that, these regions I can activate acquisition or bleaching or analysis, whatever I want to do. And then the last tab down here is important for you to activate. So you want to activate autosave and make your own drive. Here it's a local drive. Just put all the files in my folder. So this is actually where you start. And you give it some name and save it as a file format you want. And I recommend you to use a Carl Zeiss image, because then you have all the metadata with the image. You can always afterwards change to a different file format if you want. Of course, you can also use, for instance, JPEG if you only want to have some simple images to be able to send it to via email or something. Just as long as you know you are losing a lot of data doing that. And in the file format, or you can separate the files, and you can make directories in this lower tab here, so you can keep track on your files. In this tab you can just see the focus, and you can go to work position, load position, and the same functions you have on the microscope. And here you can change, activate the autofocus mode, and the stage control. You can set 0 and change the step size and so on. So instead of using the joystick, you can do it all on the screen. You can add, if you have this position as starting point, you can add this as a 0, and you can manually move around, and then you can always go back to that position. So see, going back to 0. Then you can see different processing functions here that you can do on the imaging PC number 2. And you'll probably not sit at this system to do processing, so I will not go into details in this video with this, but you can see some functionalities here. In the maintain tab, it's mostly for me, facility users, so please don't mess up anything here. Yeah, and I'll go back to acquisition and move my files and be ready to clean the system and stop it. One more thing, when you have a good image, you may want to reuse it for next time. So you can simply load, let's say this image was my perfect image, and then I press this reuse button, and you see all these things you aligned, it will pop up. So you will save some time, and you will also make the same experiments comparable. Up here you can, of course, manually save. So after imaging, you want to go to load position, and we're about to clean the system and shut it down. So first I take down the illumination pillar, and in load position I can readily take out my sample, take a piece of filter paper, or put on ethanol, and then I clean the oil using circular movements, not to scratch the objective lens. Also, of course, your sample, you can clean it. Then you close down software, and you can take the laser to off position. You go inside the next room and turn off the laser. So it's important to turn off the laser, and then after cooling down, you can turn off the whole thing. So after you turn the key, it needs 15 minutes to cool down. And after 15 minutes, you press this button. So you can, while waiting on the laser to cool down, of course you can work on the PC and move your files or whatever you want to do. Otherwise, shut down the PC and shut down the components. And then don't forget the red switch shutting off the real-time controller. And after 15 minutes you can take off the power of the laser, and you are completely done. Of course, remember to cover it up, close it, and write in the lab book.